Hi, everybody. So, what I want to do right now is just offer up a different way for you to look at the hemoglobin oxygen dissociation curve. Um, and that's in the form of something called Le Chatelet's principle, which relates to chemical equilibrium. It's something you're going to see in AP chemistry if you decide to take that. Before I show you this different method, I just want to go ahead and remind you of the two different states here. We have the situation in the lungs and the situation in the tissues. Okay, and let's go ahead and draw the dissociation curve for both. Here we're mapping the amount, and in the amount, since it's a gas, we're going to talk about the pressure, the partial pressure, because there are other gases. O2 is just part of that. We happen to use it for Krebs cycles, so that's why we're tracking it. And over here, this is how much O2 is connected to hemoglobin. So I'm actually going to draw it like this. HbO2, HbO2, and you're going to see why in a second. So we know that hemoglobin is this quaternary structure, and it exhibits cooperative binding, where you bind one, and then another one binds, and it helps it bind, and then another one binds, and it decreases the ability to bind. And that is so we can have delivery in this region in the tissues. Okay, and we can have binding in the lungs in a very flexible manner. We don't want it to bind too hard and not let go like we see with myoglobin. So what's happening in the lungs? Well, in the lungs there's a lot of oxygen, so we're going to draw that in red. So we have a ton of O2, and whenever we have a ton of O2, we don't have a lot of CO2. And when we don't have a lot of CO2, we don't have a lot of H plus. And when we don't have a lot of H plus, we have a high pH. And that should be a lower P, sorry. And because there's uh, all this oxygen here, in this case, is now going to be bound to the hemoglobin. Okay, we're in the lungs where it's grabbing onto the hemoglobin. We have a drop in temp, and that's just simply because the cells in the lungs aren't doing as much Krebs cycle, so we have less activity. All of these things are going to be noticed by the brain, and then the brain is going to shift this curve in the direction of binding more hemoglobin to oxygen. Okay. Conversely, in the tissues, what we have is we have less O2, okay, because we're not in the lungs. We're at the end of the distal end of those capillaries. And when we have a drop in O2, well, where is that O2 going? That O2 is going into our cells. Pyruvate is shuttling it into the mitochondria, and we're producing all that CO2 and all that ATP. Okay. Well, if you got a lot of CO2, that CO2 is now going to notice water. It's going to bind to water. It's going to produce carbonic acid, which is going to produce more hydrogen ions. And wherever we have a lot of hydrogen ions, we have a low pH. But remember now that the oxygen is being delivered to the tissues, it's jamming it through Krebs cycle, producing a bunch of CO2, doing all this electron transport. So that's just a lot of metabolic activity, which is going to increase the temp. So whenever the body, in this case, notices, when the brain notices a drop in oxygen, or an increase in CO2, or a decrease in pH, or an increase in temp, the brain says, okay, you are at the tissues, we need to help you drop off O2. And it's dropping off O2 because if you notice, let's say at this point, where at once this much hemoglobin was bound, now there's less. Okay, so it changes the conformation of that quaternary structure, dropping off O2 at the level of the tissues. Okay, so that's fine. That's right. But in this region right here, and I want you to be doing this in your online notes for Unit 2 for protein structure. Okay, let's look at this through the lens of a chemical reaction. Okay, and the chemical reaction is a real simple one. We have the red blood cells, those like, 500 billion hemoglobins or whatever it is. Okay. And those hemoglobins right here have the potential of grabbing onto oxygen and making HbO2. Okay, and actually there's 4 oxygens here, but we don't need to get into that right now. So here's the reaction right here. We have hemoglobin binding oxygen, okay, and carrying it around through the body. So this form right here, we call this deoxyhemoglobin. And here we call this oxyhemoglobin because now the oxygen is bound to the hemoglobin. So we have this really interesting situation that can happen where we can predict the direction in which this curve is going to shift based on the chemical reaction itself. And the idea is pretty simple. If you add an ingredient, so let's say the amount of oxygen went up, like in the lungs, okay, then the reaction is going to shift to relieve that strain. So if you added a bunch of oxygen, the reaction is going to shift in this direction. Okay? It's going to shift in that direction to get rid of that oxygen and bind it to the hemoglobin. One way of thinking about this is just a little trick in my mind. Up, up, and away. So if you add a bunch of an ingredient, the reaction is going to shift away from that increase. And it just makes conceptual sense. If you add an ingredient, the reaction is going to make more of the product. Add an ingredient, make more of the product. So this is the situation in the lungs. Okay? In the lungs, we get a ton of oxygen. And if we get a ton of oxygen, the reaction is going to shift to bind more of that to hemoglobin. And that's where we see right here. Okay? So this would be like an increase in oxygen right here. Now, conversely, what do you think would happen if we drop the oxygen? Well, what's the reaction going to want to do? The reaction is going to want to relieve that strain. And what is the strain? The strain is losing the oxygen. So what's the reaction going to do? It's going to try and replenish the oxygen. So this reaction is going to shift, letting go of hemoglobin and oxygen and producing more free oxygen, right? And this is the case in the tissues, which we see, right? I'm going to draw this in green so that it's more clear for everybody here, right? So if you drop the oxygen, the reaction is going to say, okay, we now don't have enough ingredients. Hemoglobin is going to let go of oxygen. And this is what we see in the tissues, right? And that's the decrease in oxygen right there, right? So conceptually, let's look at it again. If you have a lot of oxygen, a lot of ingredients, right, that oxygen now is going to bind hemoglobin, okay, like we see in the lungs, and this reaction is going to shift in the direction of producing more oxyhemoglobin. And that makes sense. We added a bunch of ingredients, we got to get rid of them, okay? And that is equivalent to thinking about the sigmoidal curve shifting to the left so that more HbO2 is produced. Conversely, if we lose the ingredient, right, we lost a bunch of oxygen from our cabinet, well, we got to go shopping and get more. Where are we going to get more from? The reaction is going to shift in this direction, losing oxygen, okay, and delivering more of it to the tissues, and that's what we see right here. So in areas where there's a lot of oxygen, we're going to get more bound to hemoglobin. That results in that left shift. In areas where there's not a lot of oxygen, we need to replenish it. So we're going to let go of oxygen from hemoglobin. That results in that left shift here and a right shift in that curve. All right. So hopefully that was a helpful way for you to see it. And I know I was kind of rambling a little bit, but as long as you have this down in your notes, we can really refer back to that later at some point.